Okay, we will get started. For those of you on Zoom, I'm using my phone for the microphone again. Hoping you can hear me. Can you give me a quick yes on the chat if that is true? We'll see. I'm going to start with, we'll see what happens. I've got 11 participants. All right, I hope you can hear me. Welcome. Welcome back to the cold. It feels like a freezer out there again. So, this next chunk of material is something that was once part of my lecture material, and then I kind of scrapped part of it, and I'm bringing it back. So there's a handful of details that we want to think about as we move through, some of which I hope is some review material. We will see. There are definitely some concepts in here that really are important when we think about metabolic pathways and how we go about getting energy and how we use the components, the building blocks to produce other macromolecules. And it doesn't matter if we're talking about bacteria or if we're talking about ourselves. The processes that we're going to see are very similar or the same. There are a few mechanisms that we will kind of look at briefly that really are only happening with some microbes. But a lot of the focus is happening in all sorts of living organisms. And it becomes important to think about some of those concepts truly with microbiology or bacteria to give us the opportunity to understand how these mechanisms are really working. It's pretty easy to manipulate bacteria and to potentially get an understanding about some of these processes without, jumping over and thinking about people. It's easier to do some with the method and then extrapolate that to humans. So, lots of ideas when we speak about some of the potentials for bacteria to survive, what they might be feeding on, all sorts of things that truly are varied. And we've talked about some stuff before, like speaking about where microbes might be, or where you think there's no chance they could be. And likely, there's going to be somebody that's living in those circumstances that we think are just flat out impossible, right? This acid mine drainage, there's potential for microbes to thrive, maybe not thrive, but survive. It kind of depends on the circumstances, but I feel like some that might find this type of an environment or habitat suitable really might thrive in these conditions. So I say something that we think of as maybe an environmental disaster or a potential for something like that, maybe a super fun site as we see all sorts of contamination. Some of this may be the result of the microbes doing their work in that habitat in the first place, essentially creating stuff like sulfuric acid as they are metabolizing different components, sulfurs, or sulfides, ending some of these shifts. And that in turn, as we think about this acid drainage, has real here we've got some microbes living in these nodes and some roots and legumes have the potential to harbor bacteria that are able to take atmospheric nitrogen into and convert it into ammonia compounds that are usable for the plant. That, atmospheric nitrogen really is something that can't, in a living system, utilize. We breathe it in and breathe it out, right? But microbes have the potential to fix it, to take it out of the atmosphere and convert it into usable molecules for those plants. The same microbes, but other microbes can take nitrogenous waste and complete the nitrogen cycle as they denitrify and basically release nitrogen gas back to the atmosphere. So it cycles through. And at the tail end of this chapter, cycles that we will look at briefly. Nitrogen being one of them. And just really looking at the nodes and the effects that these arrows are pointing at. So, when it comes to metabolism, I oftentimes really am thinking about like the breakdown of stuff, getting energy through molecules that either I've consumed or thinking about some organism feeding on something. We grow up all sorts of bacteria on petri dishes. We'll inoculate into some test tubes with different types of sugars and see, can they grow, can they not? Different ideas for some of these processes. 1/10 or 1/2 of the metabolism, right? Really, it's the breakdown and the building of molecules. Metabolism and anabolism go together, and all of that is metabolism, not just the breakdown, right? So in catabolic pathways, you take bigger stuff, food stuff, or whatever it might be, and break them down into smaller bits. The monomers, the substances that we will flip around in anabolism and think about building into something else, right? The bulk of what we think about in these catabolic processes really is energy. Trying to get energy to fuel all sorts of cellular functions. And it doesn't matter if we're talking about ourselves or we're bacteria, right? We also are getting building blocks that potentially can be used downstream, right? But a lot of it truly is getting some energy, right? And a lot of this, when we think about the catabolic activity, we start breaking bonds that hold these macromolecules together. As we start breaking these covalent bonds that are the high energy bonds, holding these individual monomers together to make a, some sort of bigger polymer, bigger structure, whatever it might be. Those covalent bonds are super energy rich. A lot of potential energy trapped in those covalent bonds. And if we can break them, we can potentially harness that energy. And really, that's what we'd like to do. And we do it in a handful of really enzymatically governed steps to slowly break these molecules apart. And in the process of doing that, we try to trap as much energy as we can. If we just completely exploded these molecules in one step, you would have a huge release of energy, but most of it we wouldn't be able to harness, and we'd lose a lot of them in the form of heat or potentially, at the demise of the cell as it exploded in that process. So we slowly pull these molecules apart into their respective monomers, getting little bits of energy in this process. That's the idea. With the first bit is to generate some of that. And in turn, we use that energy in anabolic pathways, the building of molecules, to assemble monomers into polymers, making other things. Right? So these reactions, anabolic and catabolic, we oftentimes describe as coupled reactions. You need the two to be happening, one driving the other, truly, right? As we think about it, and we will see more details about that as we progress through, right? So here we can see ATP, the energy molecule that we think about driving cellular reactions, right? And really, when we think about these, it's the phosphates that are the high energy bonds in this ATP molecule. And when we potentially interact with them, oh man, I asked if you could hear me before. Can you hear me better now? I don't want to have to hold my phone the whole time, but I might. A little better. Can you hear me now? Okay. Okay, that will work. I will try to get a hold of the AV people. I see their number here, but when I first got here, I thought it's all good, but it's not. That makes me wonder about Friday's lecture. Probably nobody hear anything, right? Nobody said anything. You can hear me? Scratchy? More scratchy than usual? Probably. Okay. Okay. So bear with me. We'll make this stuff work. All right. Now my phone will die. That's how that really will work. All right. So the phosphate, these phosphate molecules are linked up to our to form ATP, adenosine triphosphate, right? The three phosphates. It's an adenine, a 5-carbon ribose sugar, and then the phosphate, right? Similarly, we could think of this as one of the nucleotides in DNA or RNA, right? But typically when we're thinking about them, it's just got one, right? A single phosphate, right? The ribose, the nitrogenous base, and a phosphate, right? All linked together. But when we build those DNA or RNA strands, they all come in. Every one of those nucleotides comes in as a triphosphate. And we pop 2 of those phosphates off, releasing that energy to form the backbone of those nucleic acids, whatever we're talking about, right? So these molecules are energy rich, supercharged, right? And this is what basically is going to be manipulating the energy in the cell, picking up and releasing this energy to drive all sorts of cellular reactions, right? So in its charged up state, it's ATP, 3 phosphates, and typically we snap one of them off, all right? When we think about most cellular stuff, a little bit of energy. And I mentioned just a second ago, making nucleotides, 2 of them get popped off to form the energy needed to provide the energy needed to form the covalent bond in between those nucleotides, right? That's the idea. So it gets phosphorylated, popping phosphates on, and it gets dephosphorylated, popping a phosphate off. When it's released, we oftentimes show it as a P with a little superscript I for inorganic phosphate. And it's something that another one we think about with the phosphorus cycle, being able to kind of cycle through the environment. It's different as far as where we recognize the phosphate and whatever, all right? So energy over here, solar or chemical energy, is utilized for the production of the energy needed to phosphorylate. And we start thinking about this shifting into cellular function, right? For the anabolic, the building blocks, right? And almost the instant that ATP gets charged or generated, it gets dephosphorylated. It's quickly exchanging this energy. It's constantly being charged and decharged driving all sorts of reactions, right? And that truly is what we are focusing on in these metabolic pathways as we move along, all right? So this reaction is another coupled reaction. This is like the catabolic and anabolic image that we started off with, but it's got a couple extra details in here, all right? So we can see exergonic reactions, and we can see endergonic reactions. We haven't really said what that is, but hopefully you've got some hints, right? Exergonic reactions are catabolic reactions. These are reactions that are releasing some energy. We oftentimes think of them as being spontaneous, but maybe not necessarily exactly spontaneous, but close, all right? So exergonic fit in the catabolic, and the endergonic, those take energy, those fit as the anabolic types of reaction. And in this coupled situation, we can see the red arrows being exergonic, driving the endergonic reaction, coupled, right? And there's a handful of different examples. ATP is the middleman. It's in the center of this, again, using that energy to drive reactions, right? So we're able to phosphorylate the ADP. This is an endergonic reaction. It takes some energy to put that phosphate back on there. And this endergonic reaction is coupled to stuff like metabolism, the breakdown of glucose molecules, which is exergonic. As we take those molecules and go through these processes that we're going to see in a second, glycolysis and Krebs and oxidative phosphorylation, all of these, we're trying to harness little bits of energy to, in turn, recharge those ADPs to ATP. In the process of doing this, we generate some carbon dioxide, some water, and we generate a little bit of heat in this process as well. And then our metabolic pathways, heat technically is something that's not really useful, right? We would like to stay warm, right? But generating heat in these reactions is something that's technically wasteful. And so we try to make these mechanisms function slow and steady to not release huge amounts of heat, and instead trap that energy over here in ATP, right? That's what we would hope for, right? I know this is microbiology, but a quick little tidbit of some biology to throw this in here with some heat, all right? Little kids, infants, babies, all right, they, they're growing pretty quickly. They eat a lot, they sleep a lot, and they also poop quite a lot, all right? That's what a little baby does, right? Eating, sleeping, and pooping, right? And in turn, in all of this energy consumption, they produce boatloads of heat, right? They're burning a lot of energy, and they've got some little fat reserves, some brown fat that gets metabolized a little different. And really, when we think about these processes and trying to get as much energy as we can with those little kids, with those infants, it's not really like that. And it's more heat. They produce a boatload of heat in this process of metabolism. And then as they start really kind of growing and whatever and needing more of this energy, we transition away from that and start using it for building stuff, right? But when they're little fresh hatchlings, there's a whole lot of heat that's being generated in this process. I think that's kind of... So with our catabolic reaction, charging up the ATP, which we flip it around to the other side and we think about some anabolism, anabolic reaction. Again, endergonic, taking some energy to go and build different things, like maybe taking multiple glucose molecules and linking them together, right? with bond formation to form some polysaccharides. And all of these glucoses that we've linked together, those are covalent bonds, right? Linking one glucose to the next. And I said just a second ago, those covalent bonds are super energy rich. We got a lot of energy in there. And that energy, well, we've donated it from our ATP to build these bonds, right? To fuse them together, just shuffling this stuff around, right? And we might think of some of these things like maybe glycogen in animal cells or starches in some plant cells, energy reserves that those cells have generated in the process of metabolizing, right? Maybe we store some of that away. We could in turn think about other things like maybe the production of lipids and storing other fuel reserves for later, right? Truly depends on what we're thinking about, right? So oftentimes, almost always, when we think about metabolic reactions, when we think about these processes that are happening inside of our cells, they're almost always governed by a different type of enzyme that's helping these reactions to occur. Okay? Enzymes are types of proteins that in turn either help push molecules close together to form a bond or put strain on those molecules to break some bonds, one way or another, right? Whether we're thinking building stuff or breaking stuff, enzymes typically are helping these reactions to occur. So this image shows what's up without an enzyme in green or with an enzyme in red, all right? And the idea with those enzymes is to make it easier, to facilitate these reactions. And we say basically what they're doing is lowering the activation energy. The energy needed to get those molecules excited enough that they could do something. Form a bond or break a bond. That's really what we want to think about, all right? And, you know, as I was reviewing this stuff over the weekend, I like this figure because it's showing these reactions, but we're seeing both of them, all right? We've got our initial reaction, this reaction from X to Y, and we've got our reactant to product, right? We can see our energy on the Y-axis and this reaction path. This could be time on the X-axis, right? We go from lower energy to higher energy in this first bit from X to Y, right? Reactant to product. So we are going from a state of more kind of disorder, individual monomers, to a state of more order, a bigger macromolecule, a polymer, all right? Now we've built a 6-carbon sugar or something like that, all right, at this point. These products are more ordered. They've got more energy, all right, more covalent bonds holding molecules together, all right? And in turn, we can flip it around the other way and go from Y to X. And we could look at it just in reverse, all right? Where now we could say, instead of the product or reactant to product, we could say these are the reactants going down to the product, going the opposite direction, all right? And it's not the same enzymes that are making this happen, but it's a similar idea where we've got enzymes that are helping to get this reaction to occur, all right? And in these circumstances, if we are thinking about this case going backward, our activation energy is just this little bit right here, right? From what we would be calling the reactant, right? To get up over this hump down to here. And there's a good amount of energy that we'd be releasing in this process, right? The opposite, when we are going from the more disordered to ordered, there's a higher amount of activation energy. It takes more energy to get all of these molecules put together, right? And without it, this activation energy is tremendous, potentially inhibited, right? We can't get up over that hump, right? When I talk about some of this stuff in biology, I use like a ski hill as an analogy when we think about some of this. You could go to Pebble, and you could park in a parking lot, and you could get your skis and your snowboard, and you could just walk to the top of the hill, not even buy a lift ticket. You could just walk right up there, and you could go down the hill, all right? But I bet you're gonna do it once, right? If you even make it to the top of the hill. If it was me, I'd get about halfway up there. I'm like, fine, I'm just gonna go down right here, right? Maybe not even halfway up, right? So park in the lower parking lot and just walk up the hill to get to the office to buy a lift ticket, and you're done, right? That's the problem, right? But you buy a lift ticket, that's like the enzyme, right? That all of a sudden shuttles you right to the top of the hill. It takes you hardly no energy to get on that chair and ride to the top, right? And now you might go down a dozen times, right? Versus the one time that you did walking up by yourself, right? The activation energy gets dramatically reduced with an enzyme or with a lift ticket, right? That gets you up the hill much quicker than walking yourself, right? That's the idea. And as we look at this, all it's doing is lowering the hump, all right? What we are getting out of this, either the energy going in or the energy coming out, one way or the other, building or breaking, it's the same, all right? We're not changing any of that. All we're doing is making it easier to happen, all right? Lowering the activation energy, speeding up these reactions, whether we're building something or breaking something apart, right? Even those reactions that we say are spontaneous, you technically need to give them a little push. You need something to help get that to start, right? That's the idea. So there's a lot of ideas as we move through here. This green glob down here is our enzyme, and the gray, and the green bit up here is the substrate. The substrate is some molecule that can interact with this enzyme. And like I said, we may put strain on it, so maybe two individual monomers, pushing them close together to form a bond, or maybe one bigger molecule and putting strain on it to break that molecule into bits, all right? One way or the other, all right? And in this case, we're going to break this one apart, all right? So if the substrate interacts with the business end of the enzyme, we call that the active site. It gets in and interacts kind of like a lock and key. We say it's got an induced fit. So once the enzyme and substrate match up, you get a conformational change. The protein in that enzyme, that protein shifts the Lilith fit. In this case, putting some strain to try to break it apart. The substrate gets converted into product. They get kicked out of the enzyme, and the enzyme's basically back open for business, ready to grab more substrate and produce more product. The enzymes facilitate these reactions, but they don't get used up, right? They just keep participating, right? They are proteins. They potentially get damaged or altered, maybe denatured. They may need to get replaced, but it's not something that is used up every time it catalyzes a reaction, right? Recycled, right? Oftentimes, when we think about these chemical reactions, the product of one reaction becomes the substrate of the next enzymatic step, and it's like a chain reaction, substrate to product, over and over and over, right? Sharing. So sometimes, maybe many times, but sometimes our enzymes don't function just by themselves. They need some helpers, right? cofactors or coenzymes help to facilitate these reactions. And they might be like some types of vitamins or some metals that help to allow these reactions to occur, right? A good number of cofactors are like some B vitamins that in turn help to get some of these reactions to occur, right? And B complex is a pretty good vitamin to give you a little bit of energy. And when we think about it as this example, if it's helping enzymes do their function, kind of makes sense that it might make you feel a little more energized because you're helping to give some enzymes really what they need to help their reactions occur as they should, right? You're helping your metabolism, right? So doesn't mean you should just eat a bottle of B vitamins for breakfast, right? That's not what you should do, right? So initially, if it has some helper molecule, cofactor or a coenzyme or both, if it needs those participants, when it doesn't have them, we call it an apoenzyme. And it's basically closed for business. Okay? It can't function. But when it does get its helper in there, now the active site is open for business, if you will, and it can do its job. And when its substance is interacted with it, coenzyme or cofactor, like I said, then we call it a holoenzyme. It's got those things, right? And this in turn might be something that makes reactions happen or not based off of the binding of these substances or the lack thereof, right? Maybe what happened here? Okay. So these reactions, these enzymatic reactions, can be controlled, and there's a couple of ways that this happens. So both of these are inhibitors, and these inhibitors can be competitive or they can be non-competitive, right? And a competitive inhibitor, although it looks different, right, than the substrate,
Hello?
part of it fits into
Hello?
that active site of the enzyme,
Hi.
just like the substrate would, all right? And this is kind of sort of an issue because it looks completely different, all right? But a bit of it would look like that substrate. And so when it competes, the competitive inhibitor is competing for that active site. And if it can get in there instead of the substrate, well, now that enzyme is tied up. And it feels like it's trying to act on the substrate, but it's not. It's the inhibitor. And it doesn't do whatever function it was supposed to be doing because it's got the inhibitor in there, right? Maybe slowing that step down, right? Maybe that's good. Maybe we've got a boatload of products that's accumulated and we don't want to make any more of it. So the inhibitor would stop that reaction. Maybe the product drops off and in turn, now we need to make more of it and the inhibitor gets kicked out of there and we start going, making more stuff. Kind of just depends, right? But it's competing, all right? And in these instances with competitive inhibitors, the concentration of it and the substrate for it to function really properly is about the same amount of inhibitors as substrates, and that in turn causes these things to function properly, if you will. Right? With the non-competitive inhibitor, these guys are binding at a completely different site on that enzyme. We call it an allosteric site. This is like, maybe kind of think of it like an on-off switch. The inhibitor links up to a different area on the enzyme, and it causes the active site to switch its shape up, basically closing it for business. And now the substrate can't bind even if it wanted to, right? It can't fit in there. And in these instances, the non-competitive inhibition, the quantities or concentrations of those versus the substrate can be much, much less. Right? We don't need a boatload of inhibitors to compete for that active site. We just need a few to shut those switches off, and in turn, interacting with these processes. Some of these circumstances, when we think about this inhibition, we will call feedback inhibition, where it's typically like the end product in a chain reaction of events. That end product will go back to the very first enzyme in that chain of steps, and it will inhibit it one way or another, all right? Oftentimes non-competitively, all right, at an allosteric site, shutting it down. But it just kind of depends as far as what it's doing, all right? So not only can they be inhibitors, but there also could be some potentials for some activators. And these activators link up at an allosteric site, just like the inhibitor would, but in the opposite. When that activator comes into that allosteric site, it causes that enzyme to have a conformational change and make the active site open for business instead of closed, right? Turning it on instead of turning it off, right? And the bottom bit is the feedback, where we go through these series of reactions, the substrate to the product, so on and so forth, till we get to this end product, the square, right? This yellow square can go back to our very first enzyme, and it inactivates it allosterically, right? So non-competitive. As it links up there, it changes the shape of the active site, and now our little substrate can't link up in there because it doesn't fit like it should, right? When the product, the yellow stuff, when it gets depleted, it typically switches this back on as it falls off of the enzyme, and we're back making more stuff as we move through that. So this, as we shift gears the littlest bit, we want to start thinking about metabolism in detail, right? And it doesn't matter really, biology, micro, whatever. When we start talking about these processes, the focus, we always start with sugars, right? And we think about glycolysis and citric acid cycle, and then some electron transport or oxidative phosphorylation. We think of these as the steps and discuss how we make energy, right? But we're consuming more than sugar. We think about other things that also get manipulated and utilized in these processes. And we also think about some of the molecules that we are generating in these steps that can be precursors for other things. So we show these things, and we'll show going all the way through, producing boatloads of energy, but oftentimes we're not going to go all the way through. We'll get little fragments, we'll get pieces, monomers, building blocks for other things, and they will get shuttled off for some anabolic pathways, right? It truly depends on the circumstances and other things you're consuming, right? More than just sugar can be metabolized and can be manipulated, and those building blocks get used to build other things, right? So it's not just sugars, and we will touch on some of the other stuff in a bit as we move through here. But again, we got to kind of focus on this. And truly, when we think about these biochemical pathways, this is one of the main arms of the biochemical pathways, and other things settle into it or out of it. So it really kind of makes sense that it becomes the big focus, right? So glycolysis, there's a couple of ways we can break some sugars apart, and this is one of the main mechanisms. M. de Meyerhoff-Peranis, 3 researchers way back in the day that ultimately figured out these steps, right? And there are numerous things. I opened up OpenStack. I don't know if I'm going to toggle over to it today or not, but there's a couple of things in there that in chapter 8 that I thought were good, a couple of tables in none of those tables got put into the PowerPoint. I don't know why, but they're not. And I've tried to rip them off from the open stacks and slap them into the PowerPoint, but they don't really participate like they should. So typically share that stuff. But there's other stuff that I found. I knew was there, but I didn't really look at it. In the text, in OpenStack, at the end, in the appendix, it goes through some details of this that are really pretty impressive, all right? Deep down, a lot of what we're showing in this is really, I feel like, sufficient. But there's a lot of stuff that we kind of gloss over. Lots of enzymes, lots of steps that the appendix spells out and shows in pretty nice detail what's happening, all right? And it talks about more than just Embden-Meyerhof. We're going to talk about some as well. But, again, this is the main one, all right? And when it comes to us, this is what's happening in us, all right? Boatloads of microbes do this as well, all right? And then there are some microbes that can do some stuff a little differently, all right? But that's really why we start with this, because it's pretty much something that we see happening in almost everything, all right? That's kind of the idea, all right? So, not exactly the same, but a similar sort of an idea, like that enzyme that we were showing, that figure with the energy and the activation step, the activation energy, lowering that with the enzyme, right? That little hump you got to get up and over, right? The breakdown of these molecules, we say this is an exergonic reaction. We're going to release a lot of energy, but we technically have to get the ball rolling initially. It takes a little bit of energy to push that ball, if you will, right, to get it all started, right? So the first bit of glycolysis is an investment stage. We have to put some energy into the system, and then we're going to get more out of it, all right? A little bit to start it, more once we get going with it, all right? So it takes two ATP molecules, right? And these ATP molecules are going to phosphorylate the glucose. They're going to donate phosphate, right? They're going to give some of their phosphate to these sugars, right? And they don't care because they know they're going to get it back, right? So it happens in a couple of steps, right? A couple of different enzymes and some modifications, right? And I will probably flip that other picture up there so you can see just a little bit. But the idea is one phosphate comes into one side of the sugar, and then through a couple of other enzymatic steps, we rearrange the molecule the littlest bit, and then we pop one more phosphate onto the opposite end, right? Now we've got two supercharged phosphate molecules on both ends of this 6-carbon sugar, and that puts a huge strain on this molecule, and it causes it to split into two. We break it in half, right? So once it's got both phosphates on it, it's called fructose diphosphate, and it splits into two 3-carbon molecules with the phosphates attached to them, right? And we call them glyceraldehyde 3-phosphate. Technically, we are getting one molecule. They look exactly the same, right? Technically, one molecule is kind of the opposite, right? It looks different. Phosphate on the other side, right? And it's dihydroxy. I can't remember. Different, right? But it gets manipulated, right? And flipped around. That's why I need that figure up there. There's so many different names, so many enzymes. Doesn't matter, right? The end goal is to create two 3-carbon glyceraldehyde 3-phosphate, right? We're going to manipulate them the littlest bit. Here's the payoff phase. and we generate the 3 carbon molecules, pyruvate, all right? Popping those phosphates off, all right? Recharging those ADPs to ATP, and we call this, as this is happening, substrate level phosphorylation, because we're getting it from some molecule, a substrate, all right? Not oxidative phosphorylation that happens at the tail end of this stuff, all right? In the electron transport chain. Different, right? So what we generate in this first bit of glycolysis are some electron carriers. Our NAD pluses get charged up with their hydrogen ions, becoming NADH, and we get some ATP molecules. And we see both sides of this, right? NADHs and ATPs that we generate in this process, right? One of the phosphates coming from our glyceraldehyde, and another one in the process of harnessing these hydrogens takes an inorganic phosphate and recharges an ADP, giving us 2 on both sides, right? So there's 4 total ATPs that we get down here at the bottom, but it took 2 to get the ball rolling. So we say glycolysis, we get a net 2 ATPs out of this reaction, right? And with that, this process, and it, like I said, this is with lots of stuff doing this, right? Glycolysis is what we call a universal pathway. Everything can do glycolysis, right? It doesn't matter if you're a plant or an animal or a microbe. It doesn't matter if you breathe oxygen or if oxygen is toxic to you. Everything can do glycolysis. What you do after that, well, now it matters, all right? But before that, everybody can do this, right? And maybe all you can do is this, and you generate a little bit of energy, not so much, right, but enough to survive, right? If we can break it down further, now we can actually do something. We can do something about it, right? That's the idea, right? So here we can see our phosphoenolpyruvate, pyruvate kinase is going to basically, in this process, we are going to take the phosphate and put it onto this. So this, again, substrate level phosphorylation. This PEP is our substrate, and we're going to take that phosphate off. Kinases, there's all sorts of different enzymes, and a kinase is a type of enzyme that manipulates phosphate, either putting it onto something or taking it off, and either putting it on. But in this case, settling it from this substrate to this molecule over here. ATP gets charged up, right? That's the idea with basically this first bit of ATP production, right? And we will stop with that. With the idea microphone stuff. And you can hear me better on Wednesday. Farewell. If you haven't finished this yet, get on it. You could start it at 4:45. You'll be fine. No. Oh, maybe. I don't know. I don't know how long it'll take you. I would think you would need more time than